I forget who. But they're saying that navigation was working too well and the ROVs could move too soon. They preferred much deeper dives. Where's the excitement? The challenge.
Bridge, more to see up here. Those places.
Yeah. 30 meters, bearing 280.
Lots of cool shell hash and stuff going on around here.
Mm-hmm.
We're working with the Santa Barbara Natural History Museum. We have Daniel in the science chat with us, and he is collecting scoops of shell hash. He's looking at micro mollusks on the scale of 0.5 to a couple millimeters in length.
Is he comparing the micromollusks here with other collections that he has from different parts of the basin, or?
I think he could be. I think he's trying to expand his catalog. So he was just asking for these grunge shell hash samples from anywhere we could get them. And we did find a hashy area earlier in the dive, so we took a scoop there.
And this is Daniel of the punk rock mollusk fame.
It's kind of hedging your bet because you only have one scoop. So if you get it early in the dive, at least you know.
That's right, Nev.
We've got a basket star coming into view. Oh, and Daniel has a great suggestion. If we have excess cores, we could also use that to sample grunge.
Bridge, Nev?
Yeah, let us know, Daniel, if you have an area that you're really interested. We can try to take a
meters,
sediment core
bearing
sample for you, see if
280.
we can get the shell hash in the top layer.
Is that basket star you noticed earlier, Lizzie? Not hearing me.
We can hear you, Julie, but I suspect Lizzie might have
Okay.
dropped out. I don't think she was hearing us.
Okay. Yeah, that's happened to me a couple times too, where I'm like, oh, it's crickets.
Yeah.
We just lost her. And she just came back.
Were people saying things? I think my V-Link did that thing again.
We're talking about you.
Yeah.
Oh, I noticed that basket star, the crinoid species. I was wondering if that was the one you saw, but I didn't hear back from you.
Oh.
It's okay.
It happened.
Do we want to look at it? Basket star? Is that what we're doing?
No, I was curious. We can keep going.
Decorator crab.
Mhm.
Oh, yeah.
This thing. No. It's not. Zoom in.
Another crab.
Al.
I think I heard you say something, so I'm going to zoom.
I guess he is.
Any cup corals?
No, I'm looking.
Munching on it.
Oh, here comes something. That's not a very big rock.
Okay. We done with this guy? Moving on. We gotta go. Zoom out.
I just like that it has a self-growing clothes
Mhm.
on its body. I mean, you have to think that the sponge and those zoanthids will continue to propagate. So it's not like he has to find more material.
Now that's a Halloween costume.
Yeah,
Just little bits of whatever you have around the house.
self-propagating
Yep.
costume.
Just wear duct tape all over, so whatever you touch that day just ends up sticking to you.
Oh boy.
Actually, their carapace material is kind of has more of a Velcro-like texture if you look at it under a microscope. So maybe wearing a suit of Velcro is more appropriate.
Know what I'm talking about.
See, the crab has a Velcro-like texture.
Mm-hmm. Yeah, little hooks all over its carapace.
Oh, the hooks, not the loops.
Correct. Yeah.
Seems like a lot of work, and then they molt and have to start all over.
Mm-hmm. Pretty incredible.
Or I think what we could go be as hot for Halloween costumes would be bagpipe sea spiders.
Oh no.
And then you just play the bagpipes every time you take a step.
Oh no. That can be Paul's
My
costume.
mom texted me. My mom texted me. She's like, I heard the bit about the bagpipes. I was like, oh, that's that. Is that all you remember?
Bridge Nav?
We were talking, Julie, about sea spiders, like terrestrial spiders, can breathe through their legs. They respire somehow through their legs, which I did not know that until last night. Or the night before.
That is-
I'm not sure which dive.
That's a
that was.
nice piece of trivia. Yeah. So you're calling them bagpipe spider legs?
Well, because I was like, well, can you imagine what that would sound like? And the only thing I could think of would be bagpipes.
Horrifying.
Right. And then I was like, well, let's take this one step further. Imagine yourself at the bottom of the seafloor and it's pitch black, and just in the distance you hear some bagpipes and they're getting closer and louder. But you can't see anything.
I don't know what happens when you play the Jaws theme song in bagpipe.
Yeah.
Deep sea horror stories.
You get thrown off the ship.
You know, my son has to write a suspense essay that's due tomorrow. Maybe I should tell him about the bagpipe spider.
That's it.
The deep sea habitat.
You heard it first here.
Zoom in out.
Same coral.
Oh, yeah.
Steve, when we collected it and the polyps retracted, you made a sound that perhaps suggested you
Okay,
thought
zoom out.
differently about your ID.
Not all the way.
No, I mean, so
Yeah, that was good.
this group is, the plexaurids is complicated. Very rarely can you identify it beyond family. You might be able to get to genera in certain circumstances, but it does have some fairly distinctive morphological characters that we can look at when it gets up on deck. But in reality, if you're doing a real morphological ID, you would need to do a sclerite analysis, which would involve dissolving a bit of tissue in bleach, and then looking at the sclerites that fall out of the tissue.
Sclerites.
Yeah.
Interesting.
And then, or the other way is just to sequence it. If you have the time and patience and tools and money to do that, get a DNA barcode and you can try and compare it that way.
Yeah, the bleach sounds more accessible.
Yeah. Although it's sometimes more inconclusive. There's a lot of
Oh yeah, I'm
there's
sure.
a lot of shared.
You get what you pay for.
Yeah, there's a lot of shared traits in sclerites in this family, especially in certain clades within the family, which make it difficult to identify genera and especially species. And it's actually one group that is top priority for revision, most likely in the next, hopefully soon, because it's badly needed in certain genera.
Cool. So will that sample contribute to that effort eventually?
Yeah, it could.
Wow.
Yep. The centers for
It's all worth
diversity
it.
for the deep water Plexauridae are the tropical Western Atlantic and the Central and Western Pacific. So you find most of the species in that area, but the Eastern Pacific does have a fair amount of species as well.
There's another one of those, was it spotted or striped? Striped ratfish? Rat tails? Ratfish. Shoot. I always
Oh,
get them mixed
I
up.
thought it was a spotted ratfish. I was looking them
Spotted.
up.
I mean, it looks spotted, so I believe that. It has a little stripey on it too, so wasn't sure.
Yeah.
Is that a cup coral there to the right of that rock?
The little guy. Zoom in.
Is it on something we can pick up? That's the next question.
It's just trying to establish itself.
I know. Well, see, and that's
Zoom
the
out to
interesting part, is we were looking at where all those other cup corals were, and then this one is just out here in the sand.
Ready?
Yeah.
All right, going in. It's the end of my lens.
That's good, because that's the end of my pan and tilt.
Yeah, that's the one we want, but it looks like the only one. I don't see any.
You want more than one.
Yeah, we'll probably want 5. So she
From
has enough.
The same place?
Same general area, yeah. And also just to make collecting easier, because it can be kind of difficult, and you wouldn't want to collect one and then have to search for more.
Yeah. Okay.
So,
Zoom out.
thank you.
How far you want to go?
Just a little bit more. A little bit more. Yeah, okay.
Okay, so now we're looking for needles in a haystack, not just one.
We know they're here, and they're very orange.
That looks very orange.
Steve,
Yeah.
you've got a illustrator back there. How about you get your orange pen going?
I just draw some cup corals. There they are.
Cartoon
Found him.
corals.
You can do emojis. You can
Yeah, what's the cup coral
Yeah,
emoji?
there's no coral emoji yet.
Yet.
No, I've got crabs, shell, shark, squid, whale, lobster, octopus, fish,
Got an orange.
and orange.
Yeah, that'll
Yeah.
work. The orange emoji.
Yeah.
More or less the
Okay.
right shape.
I'm
Alright,
not
still
feeling,
bubble
I'm
cam
not
for a
feeling the
sec.
Telestrator today.
Yeah, take it. Run with it.
Not feeling this telestrator. Are you doing okay, Steve? Have you been processing too many samples? Oh no. That's the other cup coral.
Those little tiny starfish, they're everywhere. Like multiple species of little tiny starfish.
I know. I'll start keying them out.
I guess that means at least they're doing well if they have such small recruits in this area. Considering sea stars in general weren't doing so well so many years ago. Or not that many years ago, just recently.
Yeah.
I don't know if it affected or
I
how
don't know
deep
if I was just thinking that because
went.
it was, yeah, it was definitely intertidal, subtidal, and it affected the bat stars and it affected the ochre stars
Like the
and
Pisasters, yeah.
the Pisces, yeah. But I don't know about this deep. But we are seeing recruits of those different species coming back, which is good. But it was
Yeah.
a devastating event.
Got a nice little sponge there. That little tall sponge on the right. More species of different kinds of cup corals, the flabellum. Lots of little hydroids and encrusting sponges. Encrusting, I guess not encrusting, but maybe colonial tunicates. Lots of good coverage, live coverage on these rocks.
Okay.
Snails.
Good.
Yeah, we've got grazers with some of the the decorated crabs, and we've got the filter feeding, and even the stars could be doing some either they're feeding off the algal film or they could be predatory too.
Is your lens extended, Bob?
Yeah. Like maybe.
Oh, that's a cool fish. Not sure what that is. Oh, there's
What kind
two.
of fish is that?
Where are you looking?
Right under the lasers. Two fish there. Huh.
Nice.
What are those? Perfect.
Where's Tom Leidig when you need him? Or Erica?
Mm-hmm.
Does that have a little... Is that something on top of its head, or is that just like something behind it?
Yeah, hard to tell. Oh, it looks like the other one has those little feelers on top of its head too.
Oh, so they must be some kind of, is it sculpin or blenny that has those? Little
Sculpins have them.
Sculpin, yeah.
Eyelashes.
Yeah, those look like they'd hurt you.
Almost looks like a comb fish.
Almost, huh?
Mm-hmm.
Very spiky.
Yeah, like a short fin comb fish.
Trying to figure out what it was.
Okay, zoom out.
We gotta catch up.
How far out?
That's
Junk
good.
zoom?
We gotta be in close enough so we can spot these bitty cup corals.
Yeah, I appreciate that.
Am I like off to the side there? What's going on? Yes.
Look at
Oops. Catch it up.
Zipping. Ship has 2 meters before it stops, so I'll let you catch up. So science, we're about to complete a ship move, and we're just pretty close to this target we had originally dropped for the start of a transect. I'm thinking it might be worth starting it a little early so that in case this next blue area is a little bouldery, as we just saw in the last area, we're not having to stop the transect.
Sure. Yeah, that sounds great because this habitat is different. There is cobble here.
Okay.
As
perfect.
I say that, it disappears, but that's okay. Yeah.
Oh. right as it starts. From our sonar, it looks like it's pretty cobbly.
Oh, good. It was just in that moment, and I was like, I had to say it.
Yep. Okay, let's do...
What's the bearing gonna be?
We're gonna do a little different here. Let's go for... Well... Let's go 27. I guess we are staying pretty close to the ship. Well, I'll keep on 280, actually.
280.
Actually, that's gonna be the bearing.
Yeah.
What? You're at 290 right now. Yeah, 280 will be good. All right, bridge nav. (200 meters, bearing 280).
It's a cool rockfish.
Oh, do we want to come wide for the transect?
Yeah.
Yep, and you've got your lasers on.
Oh,
Perfect.
you want to zoom out? Jump zoom.
Ship move is starting now.
Roger.
Yeah, now we'll get to count that fish if we catch up with it.
And we're starting now too. Transect 10. Oh, sorry, 6. I don't know where the 10 came from.
You.
Maybe that was last night.
So we're on transect now, and for anyone who may not be familiar with that term, we are traveling 200 meters. We're trying to stay at a relatively consistent speed, but the length matters the most. And we have the lasers on, and we're going to try to stay a fixed height above the seafloor, but it looks like we're hitting some boulders, which we'll have to fly over. And the purpose of this is so we can have a rough idea of the area we've covered. Ah, there's the fish. We'll count that. And we'll count things like fish and corals and sponges that we come across. Usually anything that's larger than 10 centimeters. Otherwise it becomes kind of impossible to count everything smaller than that. And we've been using this kind of methodology, there we go, on other cruises that have been happening all along the West Coast in search of deep sea corals and sponges. And so we'll be able to use these data and synthesize them in a way where we can start looking at the whole California current in a more with a big picture in mind. But we'll also be able to use this to characterize more within the Channel Islands National Marine Sanctuary to help us better understand the resource distribution within our boundaries, and most importantly, the distribution of deep sea corals and sponges, since we have not a whole lot of data on them in various depths. And so this will help us immensely in terms of our understanding of the deep sea resources, where they are, and what condition they're in. We were seeing a lot of sponges earlier. Not a ton of big corals, but we've been seeing lots of solitary cup corals. And we're on the hunt for orange cup corals in particular, because we collaborate with a lot of different people on these cruises. We try to take advantage of the time we have and make the most out of it. So if we're going to be here collecting responsibly, we try to make the most of all those samples. So they get sub-sampled and sent to different research labs for different research questions. And we've been seeing a lot of rockfish, which is not surprising for this area of the Channel Island Sanctuary. You'll see a number of different rockfish species, which I wish I could tell you what they were, but I'm not super up to speed on my rockfish IDs, but let me tell you, there's quite a number of them, and they're all different. They're not all different, but there's many different species.
Yes, Milton Love has a whole book dedicated to rockfish and juvenile rockfish, and it's quite comprehensive.
Yes, and the title, it is probably the best titled book of all time. And I think he probably has a few books out, but the one I'm referring to is probably more than you ever wanted to know about the rockfish of California or something like that, in the Pacific Northwest.
Yeah. There's also certainly more than you want to know, and that book is like 3 times the size.
Yeah, I think that's the one I have. It's
definitely
a tome. definitely a much different transect than others that we've done.
See
Yeah.
that right there? That's an olive rockfish.
Ooh, very nice.
Every once in a while I can call them out.
Yeah. They have those prominent white spots on the latter half of their dorsal side, right?
Mm-hmm.
Greenish. And we're seeing some pretty good sized rockfish here too. We saw lots of little juveniles earlier when we were into that cobble, and now that we're into some larger, chunky habitat, we're seeing chunkier rockfish.
Yeah, it's a really nice diversity of rockfish in this whole southwest portion of the MPA around Richardson Rock.
And I just got a message from Daniel in the science chat, who works for the Santa Barbara Natural History Museum. We collected some brachiopods earlier. L. california. I'm not going to pretend I know how to say the genus name, but that particular species has not been recorded from San Miguel yet. The closest is northeast corner of Santa Rosa. And that's the only record from the Northern Channel Islands. So it's exciting that we were able to get that for him, so that can go down in the books.
That's great. So yeah, so this sort of information and data can really sort of help inform sort of thinking about range extents of different species. I know a long time ago when I used to do trawling down off at Dana Point, Bob Lee with Fish and Game used to always contact me and say, anything unusual in that trawl? Because we're trying to expand our understanding of where fish, their ranges are along the coast and subtidally and in the deeper portions. And so, yeah, I think that's what it's nice to have this sort of opportunity to be able to identify new locations for species. Yeah.
And because of the science chat, we were able to facilitate that collection because I'm no brachiopod spotting expert. And so, so we did collect them, thankfully, because Daniel's been joining our dives with us. But not only that, he was able to tell us pretty immediately, now that we've collected that sample, the implications, the potential implications for what that sample means, which is only possible because of the Ocean Exploration Trust's telepresence capabilities. Just the other night, we were off of Santa Lucia Bank, and we were seeing a ton of what we thought were skate eggs, and which some of them were, so skate egg cases, because they like to lay them on corals and sponges, and but we were seeing a couple of different kinds, and so we were talking about in the science chat, and actually multiple people texted, I think Joe Bizarro is his name, and he's a skate expert, and he was able to text everyone back from the grocery store because he just got this barrage of texts. Everyone independently thought to take a screen grab and send it to Joe, and later that night he sent us a paper that described a few of those egg cases, one of which was I believe a cat shark and a skate. So we were seeing multiple different species of eggs that night. And we were able to learn that only a few hours later. So the speed of this networking capability is just mind-blowing.
I think it's fantastic. Yeah. Especially since he was just living his life, checking the science chat, or able to provide that level of expertise on the fly. I love it.
Yeah. These rockfish are sure swimming around a whole lot, making a fuss.
Seem to be attracted to the lights, maybe, or
I don't know.
And earlier we saw
what's attracted to the lights.
Yeah, Say, and we've seen several species all together each time
Right.
we see a clump of them, which is interesting. It's not just all one or two.
Yeah, we were having interesting conversations about what that means in terms of what they eat and how they compete with one another. What niche do all of these different colored and sized rockfish fill?
Well, you know, and with rockfish just in general are such a long-lived species, and to see these nice size rockfish, I mean, this is a success story of the no-take marine reserve, potentially
Yes.
too. Just the fact that they're being left alone and allowed to grow, and especially since they
And
don't really,
reproduce.
they become more, they produce more eggs the older they get. So having a good sort of, you know, size class distribution is really important, I think, to the long-term survival of many different species of rockfish.
Definitely. And we know there's a lot of recreational fishing for rockfish within the Channel Islands. I know there are several companies that boast that like, pretty much almost any time of year you can get your bag limits of rockfish out here. It's a very, it draws a lot of people in to fish in the Channel Islands.
It does. And I think what's interesting too is that there are certain sort of oceanographic conditions that are sort of triggers for them to reproduce. So they seem to like it when we're in our colder water oscillation and in the warmer water oscillation, which is usually about what on average every 10 years. And so I remember after the last sort of warmer period of water, with all the El Ninos up to the 90s, and then when we got to 2000, the water it turned over, was a lot cooler. The kelp came back, and we were seeing amazing rockfish recruitment. And so it's important to sort of, you know, to have these sort of savings accounts of refuges for fish, rockfish, and other species to be able to take advantage of the opportunity when the conditions are just right to recruit, reproduce, and recruit. Very cobbly through here.
Yeah. They should be coming up to a more steeply sloping feature, which, as I say that, we're kind of gaining height now.
Here it comes, maybe.
Here it comes, ready or not. Here we are.
Yeah, it looks like it might be along our starboard side. Oh, we got squid again in Argus cam.
I missed it.
Yeah, it was just a quick
flash.
flash, but quite a few.
I'll drive by. We had seen kind of a larger group of them really quickly come in a Hercules, but they all kind of ricocheted off the bottom. They all just kind of landed in the mud and then inked and then swam away, and we were wondering if maybe something was chasing them, that they just sprinted for it and then crashed into the bottom. That's a cool rock with some sponges.
Mm-hmm.
So we're about 50 meters off from the end of this transect, and we'll be smack dab in the middle of the blue spot on the slope map at that point.
Got it,
There's
I see
the
it.
squid.
Mm-hmm.
Now that one looks like a vermilion rockfish.
Yeah.
Oh, this is a nice rock outcropping here too.
There's also so many different, mostly orange rockfish.
That's exceptionally true. And I'm always astounded by the folks who can actually distinguish between them
Like
from
Tom.
quick
My
IDs.
goodness.
Yeah,
Yeah.
it's
There's long sign,
it's not an
short
art form. It's just
sign,
a level of expertise that's amazing.
Yeah.
blackgill,
Yeah,
vermilion,
looking at the minutia of like just distinguishing
canary.
characteristics, it's like, oh, okay, hold still. I gotta be
Yeah.
able to find that.
Oh, there's
Right?
another
Yeah.
lingcod.
Oh, another lingcod coming up on the right. Seeing those crabs pretty regularly, the decorator crabs, and I think we're seeing a new species that I hadn't seen before yet, but we're on transect, so we haven't been able to zoom. They're a little bit smaller.
Yeah, they are. They're not like a sheep crab.
That's a new fish in the top left. There it goes.
That brown guy.
"20 meters left to this transect."
Cool.
And shoreside during the transect, Paul switched over to Herc, so we have a new driver.
Thanks for letting us know.
Welcome, Paul.
Thanks. Do we expect these features to be northeast a little bit right now?
On our starboard side is where it looks a little brighter, but we're kind of right on top of this feature. Not quite as dramatic as the last one.
Yeah.
No.
This is why ground truthing is important. So
Yeah.
I think if we want to explore this area to the north and northeast, yeah, like you said.
Yeah, that other one, it didn't look terribly much brighter, but that wall definitely came out of nowhere.
So we're still on the hunt for cup coral, correct?
Yes, ma'am.
Okay.
I think the cup coral is really our last priority. We could do another transect at some point, which I'm totally good with, but other than getting in maybe 1 or 2 more transects, I think collecting the cup corals and then seeing if we can get a grunge sample with a sediment core might be nice too.
Roger. Got about 1.5 meters before this transect completes. Oh, actually that was 1 meter. Transect complete now.
Excellent.
And then the ship has also finished its move, so if we want to look around here, we're good to go.
Yeah, sounds good.
Quite different.
Mm-hmm.
More crinoids, smaller cobble. We had passed over some pretty big sponges. So maybe we
Lots
can
of those little
go
tiny
a
sea
little,
stars.
definitely, a little bit northeast, and then maybe we could come back. But like, you know, so parallel to where we are now, but maybe a few 20, 30 meters north.
Sure can.
Yeah. Yeah, go for it, Bob.
We can do a short step north. Does that sound good?
Yeah, sounds great.
20 meters?
Yeah.
Bridge nav. 20 meters, bearing 0. And as far as ascent time goes, took the liberty of calculating a couple of different scenarios. On deck time is midnight, so at about roughly 120 meters at 18 meters a minute, it's a 6.6 minute ascent.
Slow down there.
And yeah, Bob's off SPL, but it's an additional 10, 15 minutes more to get on deck. And then for 15 meters a minute, if that's our ascent speed, it's a whopping 8 minutes of ascent.
Oh gosh, the discrepancy.
Yeah, so I'd say
what to choose.
So depending on where we land at near the end of the dive for depth-wise, I'd say leaving no later than 11:30, 11:40 would be great to give us a little room to get up.
So we've got about an hour.
11:35.
That could be the difference between an orange cup coral and no orange cup coral.
I know, I was gonna say now the pressure's on.
Shoot.
Find the cup corals.
Yeah, I kind of like the idea of going north and then coming back to see if we can't win in any more of those big rock faces to see if we can give it another shot.
Roger.
Otherwise, there's nothing in the immediate area really that would leave me thinking it could be good habitat. to do more searching. I think our eggs are in this basket. This features basket.
All right. Challenge is on. We got 10 meters left of this ship move.
And then is the plan to go back parallel from where we came?
Yep.
Yeah.
Got it.
Just kind of
Yeah, we can follow the edges of the faces or the boulders, however it works.
What's right in the middle
Generally.
there?
Oh.
Oh, it looks like a star. Never mind. Yeah, it looks starry. Orange.
You're gonna find them on the bigger boulders where it doesn't move as much.
I
So
agree.
a lot of this cobble
Yeah, they want more stationary rock.
has a propensity to fall downhill.
That's the conundrum.
Not conducive to coral growth.
And not conducive to coral sampling either.
Collecting.
Yeah.
Oh, we got a little junk on the bottom right. You want to zoom past it?
Yeah, go for it.
Oh, yeah, that's right. Hi, Paul.
All right. Ready for our eastward move?
Yeah.
Bridge nav. Uh, 30 meters, bearing 098, please.
Oh, what's that?
Oh.
Oh, line. My goodness. Where did that come from?
Is that where it ends?
Oh, okay, maybe.
Bridge nav. Hold position. Oh, it looks like it goes that way.
What are we looking at?
There's a line here at top. Loops up. Goes up to the upper right corner. Yep, definitely a line. Let's
So
step back down.
I just came up
Well,
and
is it on the bottom? I can't tell.
it looks like it's resting on that rock, yeah.
Okay.
Well, it's kind of
And
better to
it's
keep
moving. Yeah, we
your
should.
enemy in sight, so maybe just kind of see what it does.
Yeah, yeah.
It looks like it's
Hold,
moving
we
towards
could
the
tilt up some.
right. I feel like it's.
Oh, that's the wrong way.
Oh, it
It's
gets
making
caught
a
on that
jog
rock.
around the rock.
Okay.
Yeah, it's on that rock.
So that part's okay. But
Okay.
what's going on over there?
Yeah.
Right, it's going off of that.
There's another one right there.
Yeah.
I guess it goes over, both of them come
Loops
over,
around.
loops around the rock and goes that way.
I mean, we're about to jut out
That way.
east.
So
Yeah, okay, well,
we good to go?
lateral over to your right.
Bridge nav.
Bit more centered up.
At 20 meters, 098. I'll just do some smaller steps right now.
Sounds good to me.
A little bit of current here that wasn't there before too.
Yeah.
Right when you're trying to lateral that direction. Or I guess it was pushing us before, wasn't it?
A little bit. Yeah, I
Yeah.
guess it was on our tail.
I'll drop a target for that line as well.
Danger target.
Yeah. Just in case.
What do you think the thickness is of that line? It seems like it's more like is it like a polypropylene line or? That could be monofilament.
Oh no, definitely
know.
it was a thick line.
Yeah,
It was the ½ inch or quarter, 3/4 inch, something like that.
that's what I was thinking. Yeah. So probably from some sort of a trap or
Flag
trawl
rock
thing.
fish. I know that one.
What is that fish?
The red and white bars flag.
I guess I don't want to go too far over that way. That's where the line was.
Yeah, that's fine. Once you think we're over far enough, we can just start going back the way we came too.
Roger.
Well,
Opposite.
it's actually looks like it might be getting
a little rocky again.
a little rocky again.
Oh,
So,
well then.
Interestingly, on this side, it actually should be, I think the slope may start again. And so on our starboard side is the peak of whatever this feature is.
Got it. Okay.
So I think originally when we were looking at the map, we were thinking the most interesting things would be at the top, but perhaps it's along the side.
Perhaps not.
This looks like just one big rock.
Yeah.
And sonar's not really showing
Much else.
tremendous amount there.
Well, at least we have this one.
What's going on down inside there?
Yeah.
What is that?
Is that an octopus house?
Those.
That looks like bacterial mat.
A lot of rockfish.
That white in there?
Shells? Could be
Yeah,
bacterial
let's zoom
mat.
in.
Shell.
Oh.
There's some shells.
All right, let's come wide.
It's an odd
I'm going to push it
shape
partial.
for a rock.
Are we,
It is.
I need to drive forward and get
Yeah.
back.
Yep. Ship's 2 meters out from a stop, so let you catch up.
Moves are pretty
Lots of little
snappy
sponges
here.
on that rock.
Sure are.
Another lingcod down there. Just passed.
Okay.
I guess I have a few meters to come down, but we might want to watch the delta as I get closer to Argus again. I don't know. I think I'm going uphill here.
Ready for a move? Yeah, the slope should be, well, you're kind of on it, but yeah, you're going up slope a little bit. Ready for a move?
Yeah.
Bridge, nav. 20 meters, bearing, what bearing on the same bearing. Thank you, yes. This is why I need to write things down.
Let us know, Abby, if you want to zoom on any of these frilly looking sponges. More of that strange weathered rock with that like hole. Almost cave looking right there. That's neat. Darn. Can we zoom in on this rock with a bunch of sponges on it?
On the right there?
Maybe even in front of us, because I was talking about the frilly sponges, which this also seems to have on it.